Hello, Andrew. What are you looking at there?
Okay, so this on the soil surface. We've got all three of the bryophyte groups represented here. We've got the true mosses here. We've got a thalloid liverwort, and we also got a hornwort, which is really kind of cool because you don't see them that often. So we've got the three major bryophyte groups right here. So this is the hornwort. There and there.
Yep, with the little horns sticking out.
Yeah, you can see the little sporophytes.
Yep.
And there's another one there with the sporophytes. And then we've got these thalloid liverworts. So we've got one that's got a very narrow thallus. So thallus is basically just means like a strap shape. And then we've got one that's got a more broader thallus, which is, I can't see any, but there's some there. So this one's probably in the same genus as the narrow one, but probably a different species. And then, in amongst all of that, we've got a moss. So it's all this green bit that's all growing through it. So, oh, look at that, there's a huge patch of hornwort through here. So the hornworts tend to look a bit darker and actually look a bit slimier, if you like. But it still is it's a thallus.
Such interesting terminology, such new things to learn, so many things to remember. It's wonderful. Thallus.
So there's a good one with the other bits here.
So which one's the hornwort?
Okay, the hornwort is this little one here. Probably this one here. And this one's actually got its little sporophytes on there, the little horns sticking up.
Yep.
So part of the way that bryophytes reproduce are with spores. There's a nice example there as well. So they produce a sporophyte, so that to actually get a sporophyte produced involves sexual reproduction. So you have a male sperm and a female egg, and the sperm has to get from the male part to the female part, and it requires water so that the sperms can swim through to get to the egg. So that's why mosses tend to occur in areas where you do have that moisture around, and it's also indicating, you know, its close association to water from when it made the great leap from fresh water onto land. So if fertilization is successful, you get a sporophyte produced and it grows up. So the sporophyte is basically parasitic on the moss plant for nutrients. It doesn't have any of its own nutrients, and then it produces these spores, and the spores require dry air so they can get blown about in the wind, and then when the spores land on a suitable surface, it'll germinate. So you can imagine how many spores are floating around all around us, and again, it'll only be a fraction that would land on an appropriate surface for it to germinate and grow into a new plant.
Yeah, get your lens, Andrew. We're gonna demonstrate what it's like to be a bryologist by having you do the bryology pose.
The typical bryologist pose. Yeah, it's pretty cool.
So if you want to be bryologists, guys, you got to be able to get into this pose on all fours. And earlier we've seen people demonstrate to go down even further, just full prostration on the ground, no knees required.
That's right. Got to get down and have a look at them. Get down to their world.
Yes, very small little world, beautiful little world.
Yes.
And your magnifying lenses are of different types as well. You've mentioned earlier where we have 10 times, 20 times, and even greater than that, or you wouldn't require that much?
So the lens I've got is the general one I use is 10 times magnification, and I like this one because it's got a little LED light in there. So usually I'm working in dark areas like rainforests or things like that, where you don't have a lot of light to play with, so having something with a light source is really handy. And if you wanted to get down, you could actually hold it over your camera lens on your phone and have it as a bit of a macro as well. I also got some higher magnification ones, 15 times 15 is useful. Again, usually I have both of them hanging around my neck. At times 20 is probably getting too high in magnification. If I have to use it by 20, I usually will collect something and take it back and look underneath the microscope. So yeah, just a by 20 and one that has a light source. So a lot of gem supply shops or geology supply shops have jeweler's loops or hand lenses. So, and they're not that expensive. So, yeah.
Fantastic. Well, let's see you collect some of these babies, and then
Okay.
maybe we can chuck them under the microscope as well.
Okay, that sounds great. So yeah, really easy, pocket knives handy. So it's good to get a little bit of the substrate as well, and that's basically it. And just put it into the envelope. Now that's probably too small for purposes. I wouldn't try and fill the whole packet. I usually get around 20 cent piece size, and that's enough. If it requires genetic work, then I'll probably need a bit more of a sample that can be used for the genetic analysis, but just for a collection purposes, anything around a 20 centimeter size. Now the ideal way is to try and collect from the edges of a population as well, not from the middle, so it allows, won't disturb the center of the population. It can still expand. Sometimes that's not possible, especially when you know something like this hornwort, it's only got these individual plants that are around. So what I'm trying to do with this one is to get some of the sporophytes as well, because the color of the spores is one of the diagnostic features for this particular group. So I'll try and find some more with some of that. Oh, that's a nice specimen.
Wow.
And again, I'd try to keep things separate. So I'd have a packet for the hornworts, and then I'd have a packet for the different liverworts. But for this, we'll just collect into the one.
Okay.
So now I'm trying to get this thalloid liverwort. So this one probably end up being sent down to Canberra, because there's a bryologist at the herbarium in Canberra who's doing a revision on this particular genus for the next few years. So I've been sending her specimens from all over the place when I come across them. And then there was a thalloid liverwort, the bigger one.
So that's a thalloid liverwort.
Yeah, so they're all three of these have got it's a thallus as the plant body rather than leaves, true leaves. So it's just called a thallus, and it's that strap-like growth. So it is pretty cool that we've got the major groups all represented just
in this tiny little patch.
Tiny little area.
It's quite amazing.
Yeah.
So Andrew, tell us about how mosses are able to rehydrate and sustain themselves in drought.
Okay, well, all the bryophytes, the three different groups, the mosses, the liverworts and the hornworts, they share a number of common features, and one of the features they share is this thing called poikilohydry, which basically means that they can't control their water loss. They're sort of like in equilibrium to the water in the surrounding air. The other cool thing about mosses is that when they lose moisture, that gets to a point where they can they know how much moisture is left, and when it's getting pretty dire, they release this little protein that basically sits there, and when the water gets to such a level that photosynthesis stops, this little protein sits there until there's water available again. And if there's rain or there's fog or something like that, there's enough moisture for the bryophyte to absorb that, it'll restart that photosynthesis by initiating that protein again, which is really cool. So basically, if there's not enough water for photosynthesis, basically it'll just shut down and wait. That said, all bryophytes, they're different tolerance of desiccation. So some of the bryophytes in the arid zone can sit out there for, you know, years, just waiting for some moisture to come in. You know, things that are a bit more tolerant of the darker, more moist areas, they probably couldn't survive out in the arid zone for that long. But still, you know, these things will dry up for a couple of months easy and just sit there waiting for another heavy dew or a shower of rain to come through. The other cool thing is they've got this thing called totempotency, where that means that any part of the plant can break off and form a new plant. So if a leaf breaks off, it has the capacity to form a whole new plant just from that part of the leaf breaking off. So things like some of the epiphytic ones, if birds come along and grab some and put it into the nest, basically that if the nest is in the right sort of area, the moss will keep on growing in that nest. So the bird will have a little living nest.
That basically says that they're very important organisms.
They are.
Yeah.
They're very important in a lot of different ecosystems. So if we look at rainforests, they're very important in absorbing a lot of the moisture in the air from rain and such. So when the system starts drying out, the bryophytes, you know, are using some of that moisture for photosynthesis, but the stuff that they can't use, they're actually releasing back into the environment because of that poikilohydry. So there's that water gradient happening, and so if the air is drier, then the inside the moss plant, it's going to be losing water out in the atmosphere. Hence, the humidity, the relative humidity underneath the canopy in a rainforest will be increased, and due to partly from that moisture loss from the bryophytes. In the arid zones, they're important as part of the component of the what's called the biological crust, or the cryptogamic crust, and that's the crust that forms on the surface of the soil and basically binds the soil surface together. So it's made up of things like mosses, liverworts, lichens, algae, and basically Australia evolved. You know, Australian ecosystems evolved with soft-footed animals like kangaroos and stuff, and the cryptogamic crust basically just bound the soil surface. So when Europeans brought in hoofed animals like sheep and cattle, it broke up that cryptogamic crust, and it can lead to soil erosion either through water or wind. So very important. In the northern hemisphere, the large sphagnum areas absorb a lot of water from snowmelt, hence controlling that release of water out of the ecosystem as well. So if the sphagnum bogs weren't there, you could just get this massive water just flowing down into the rivers, but the sphagnum bogs basically just intercept all that water and hold on to it just a little bit and release it into the environment. So sphagnum is interesting in itself in that it can hold up to, like, I think it's 80% more weight in its weight of water. So huge capacity of just absorbing a lot of moisture.
Fantastic. I love these guys.
They're pretty cool.
What did you find, Andrew?
Oh, I found that Bosmbronia with its sporophytes,
Ooh,
which is really kind of cool.
beautiful. Oh, it's beautiful. All right, I got to get a shot of that. Awesome. So that's is that a leafy wort? Hornwort?
It's a, I think it's a simple thalloid liverwort.
Ah, liverwort. Where is it?
It's all just through here. So, let's see. So what we've got here is we've got a lot of things actually. I should get a pointing leaf. So we've got the hornwort is here. We've got a thalloid liverwort in here, which is Riccia. But then we've got Ortus Bosmbronia, which is another type of liverwort all the way through. And can you see those little black dots there?
Oh, yeah.
That's the sporophytes.
Oh, wow.
Oh, there's some better ones over here. Can you see them?
Yes.
And of course we've got the moss growing all through it. as well.
Such a beautiful little garden.
It is, and just an amazing diversity of bryophytes in this, 20 by 20 centimeter area. So you can see, some of the mosses can get reasonably tall. Here's a nice big clump over here. So,
Wow.
and I've just ripped out not only a moss, but it looks like a leafy liverwort there as well.
Wow, there's so much diversity.
Yeah, which is really cool.
Yeah, and especially if you know what you're looking for.
I'm pretty excited to find the Fosambronia with its sporophytes. Oh, it looks like there's possibly some moss sporophytes this here. Just in front of my finger.
Oh, yeah, that little dot.
Yep.
Oh,
It's got its
hello.
little cap on. And all the hornworts have got their horns sticking up as well at the moment. So one of the important diagnostic features determining the hornworts is the colour of the spores. So you really need to find the sporophytes in a mature state so you can see the spores still. Oh, here we go. These look like the sporophytes have opened. See how it looks? They're black at the top. So that looks to me that the sporophytes have opened up and split. Yep, there's one there that you can see that it's split.
Oh.
So that's really cool. I might actually collect some of that mature one.
So what you looking at there, Andrew?
I'm looking at a hornwort. It's got its horns on. The horns are open, so the horns are the sporophytes. I'm just trying to see if I can see the colour of the spores, which is the diagnostic feature. So to me, without having to put this underneath the microscope, I think they're black spores. So it's probably going to be what is it? Anthoceros, I think it is the genus. Now to collect bryophytes, it's pretty straightforward. You don't need a plant press. What you do need is to have permission from the custodians of the property. And if you're in national parks or state forests, you have to have the proper permits, for example. But it's not like collecting higher plants. All I need is basically an A4 piece of paper, which I fold up into an envelope. And on this one, I've got all my collecting details on the front, so I can just circle what I need to. And basically you just grab your sample, stick it in your envelope, and you're done. So that's it collected. Now when I get back to the lab or wherever I'm staying, and if it's particularly moist, like this is feeling a bit moist, I can just stand all these up on a table like this, just to let the air flow through and dry out. If they're really wet, I'd change the envelope so they don't go moldy. So the worst thing you can do with collecting bryophytes is press them, because we need to have a three-dimensional shape for identifications. And I do see a fair few bryophytes that have been pressed in the collection at the herbarium. And you look at it and go, this looks a bit weird. I don't know what it is, but it's a bit weird. Oh, it must have been thrown into a plant press. So really easy to press, and because of that poikilohydry, the plant dries out, and when you're ready to ID it, you just add water, and basically the plant comes back to life under your very eyes. So with quite a few of the mosses have these big, some of the mosses have these big cells at the corner of their leaves called alar cells. And the function of the alar cells is that when they absorb moisture, they swell up like balloons. So because they swell up, they actually force the leaf to open up and increase the area that's exposed for sunlight, for photosynthesis. As the moss is losing moisture out of the leaves, it's losing moisture out of those alar cells. So basically those cells are getting smaller and smaller. And as they get smaller and smaller, the leaves are slowly closing up. And that has a couple of functions. The closer it gets to the plant body, it's reducing how much moisture is getting lost to the air, so it's slowing that down as well. And then eventually, when it's lost all that moisture, the leaves either curl up or sit pretty close to the stem, and photosynthesis shuts down and the moss just waits for moisture. Then when there's a shower of rain or some fog, the moisture goes straight into the plant body again, swells up those alar cells, and the plant opens up again. So that's a bit of a mechanical process as well. So I've done looked at specimens that were collected over 100 years ago at the herbarium, and you add water and you can see the leaves start to open up. So the plant's well and truly dead, but because those alar cells are still absorbing that moisture, it's still doing that same sort of function in separating the leaves out, which is kind of cool.
Amazing little creatures.
They are. As the sun goes over and it dries out during the day, it just closes up again, and then with the heavy dew in the next morning, it's open up again.
What's that one called?
This is one of the Rostrolobriums in Bryophytes, and it's not the most common one. I think it's Rostrolobrium subfasciculatum, I think it is, without having to stick it underneath the microscope. But some of the Rostrolobriums, their leaves are more concentrated at the end of the stems, like a little rosette.
They're
but this
scattered along there.
this one's they're scattered all along it.
Look at them.
What have we got there, Andrew?
This is a species of moss, Hyperpterygium discolor. And they're just beautiful. I'll try and just get one plant just to show you. So it's got this type of life form called dendroid, so tree-like. So you can see it's got a little stem here, and all the leaves are sitting on top, almost like a palm tree look.
Beautiful.
And it's just likes the soil next to the creek here. And it's just a beautiful moss to look at, and it's looking much better than last time we were here. Last time we were here, it was actually fertile as well, but I don't see any sporophytes at the moment.
Isn't it beautiful?
Shot too.
Yeah. I'll take some photos of this.
I'll pass it over.
Footage
to
of this.
Thank you. What do we have there?
Hyperpterygium discolor.
It's such a beautiful growth pattern.
It is. It's a beautiful moss. This is a moss called Racopillum cuspidatum, and it's a moss that's looking a bit like a leafy liverwort. So when I first saw it sitting there, I thought, oh, it's a leafy liverwort, but I pulled it up and had a look underneath, and I could see that it's got its nerve on its leaf, so it's definitely a moss. But it's different to other mosses. Instead of having the leaves spirally arranged, they're arranged on either side of the central stem. So it's a pretty little moss. It actually grows in my backyard at home.
Andrew, so what are the proportions of mosses to liverworts to hornworts?
Okay, well, with the mosses have the greatest diversity. In southeast Queensland, I think there's around 600 species of moss alone. And then with liverworts, it's around maybe 300 to 400 species in southeast Queensland. And then the diversity drops right down with the hornworts. It's only about 10 species in southeast Queensland for hornworts. And worldwide, there's only just over 100 species of hornworts worldwide. So they don't have a large amount of diversity compared to the other two groups. Mosses do have the largest amount of diversity in those three groups.
So if we learn to identify hornworts, and there's only 10 in Queensland, then it's quite easy for someone to sort of go on a hunting trip just for those and try and find out hornworts.
Yeah, and I tend to say you don't need the fertile bits for an identification. So with a moss, you don't necessarily need to have the sporophyte present, but with hornworts, you do. You need to have the sporophyte present and have it mature so you can see what the colour of the spores are.
So it's even harder to identify them than mosses as well.
Yes.
On top of that.
It is.
So they're rare
well,
and difficult to identify.
A little tricky to ID, yeah. Yeah, you just have to be at the right time. While mosses, you know, you can collect them any time of the year and get an ID on most species. But with hornworts, if you collect them without the sporophytes, there's no chance you can get down to a species level.
So Andrew, what are these ones?
Okay, this one is, I'll just check to make sure my ID is right. Yeah. Okay, so this here is a little population of a genus of moss called Physidans. And it's actually the most diverse genus of moss in Australia. So it's got, I think it's over 100 species in it alone. So they tend to be mostly on soils, especially on creek banks or track banks. Some occur on decaying wood, and they've got a beautiful structure to them. They've got a thing called a vaginate lamina. So basically, the leaf has a little flap on the undersurface, and depending on how long and the shape of the apex is all diagnostic of what species you're looking at as well. So yeah, fascinating little group of plants.
So little. Tiny little things. They're only like a few millimeters tall.
Yeah.
Can they get more lush than this?
Oh, you can get ones that are, you know, maybe 2 centimeters long. One species is pretty big, and then others are just microscopic. They're just tiny, a couple of leaves, and that's it. It's got these other lobules sticking out the side. It's just beautiful.
What's it called, this one?
Okay, this is a leafy liverwort, this here. It's quite a large one, as you can see, compared to my finger. Now this one's called Porella.
Porella.
Porella crawfordii. So it's a leafy liverwort, and how do I know that? Because of the arrangements of the leaves. They're on either side of the stem, and there's no nerve on those. In fact, with the leafy liverworts, the leaves are called lobes. So it's got these lobes here, but the really cool thing is when you turn it over and you look at the undersurface and you can see an underleaf, and it's got these different lobules on either side, and it's just got all this complexity happening in something that's, what, maybe 4 millimeters wide.
Yeah. I wonder if we could have a look at it through the lens, and I'll try to get my camera on. Maybe it will be possible. We'll see. Look at that. Isn't it beautiful? What's the name again?
Porella?
Porella.
Yeah, I do.
Porella is beautiful.
That's true, isn't it?
Stunning, stunning. What else did you find, Andrew?
It's in a family of leafy liverworts called Luguniaceae. So I think it is Lugunia itself. So it's just this lime greeny one here. It's a little leafy liverwort.
Wow, it's so small. Little leafy liverwort.
And the thing with the bryophytes as well that occur on trees, what you might find is a species turnover from what's down here to what's up further up the trunk. Because as you go up the trunk, it gets drier. So up in the branches, especially in these closed situations, they're more exposed to wind and sunlight, and so you'll find more drought tolerant species up there compared to down here, and you're probably getting maybe a little bit more diversity down here on the trunk of this one. But this is an excellent tree.
Yeah, it's an excellent mossy tree and lichened tree. Now, you also told me here that there was another 2 species just in this little forest patch here.
Oh, okay. This little leafy liverwort here, it's quite common. It's in a genus called Frolania. And we also got this one here, which is in a genus of moss called Macrometrium. So above that lichen. So a little Macrometrium here, and then the darker green down here is a little leafy liverwort.
Beautiful.
Look at that.
Oh, it's so cute. What is it up there?
I don't know. It could be a little Frolania growing over the top of the lichen.
Incredible diversity on this tree. Small little tree with lots of lichens and lots of bryophytes of different types. So how many bryophytes do we have on this tree?
I think there's probably around 6.
Wow.
And that's just from not looking that hard, just in a space of a meter.
Talking about biodiversity.
Yes. We had rain overnight, and that's great for mosses because they come out. It makes it a bit more difficult to me because I'm used to seeing most of my mosses dry. So sometimes the mosses will look completely different from being dry to wet. So this one I'm not too sure about. So I've got an envelope here and I'm just going to gently pry it off this palm trunk. If I can find my pocket knife. And hopefully, I think it's probably related to this moss here. It's just that one there.
Beautiful. And it's fertile right now.
Yep, so it's got all its sporophytes. The thing that intrigued me was that the sporophytes again aren't extended away from the plant too much. So these are the mature ones, that's an immature one. So yeah, it'd be interesting to see what we get at. But yeah, it's just this little moss that's sitting there as well. So I'll try not to take too much, and these all end up at the herbarium collection. Not just Queensland herbarium, sometimes Canberra, sometimes Melbourne, depending on where the particular expert is.
Cool. Looking forward to finding out what it is.
Yeah, me too. So some of the important things to look for with mosses is if there's a central nerve on the leaf. So, and you also have to have a compound microscope to do an identification properly because you've got to have a look at the cell shapes underneath the microscope, whether they're linear or rhomboidal. So it is a bit tricky. It's not too tricky. You get used to it, preparing microscope slides and pulling leaves off moss plants, which is a bit like feeling like pulling wings off flies sometimes. But yeah, it's. I think a lot of botanists feel a bit intimidated working with any group that they're not familiar with, such as lichens or bryophytes. But you know, the bryophytes have a whole range of terms for the different equivalent parts of the plant, and it's just getting familiar with looking at those.